My guest today is Professor Shelly Flagel, who is Professor of Psychiatry and Neuroscience at the University of Michigan. Her research focuses on understanding the biological and psychological factors that contribute to individual differences in vulnerability to mental illness, particularly addiction. My second guest today is Professor Jacinta Beehner, who is a professor of psychology and biological anthropology at the University of Michigan. She co-directs the Simien Mountains Gelada research project in Ethiopia and the Capuchins at the Taboga Project in Costa Rica. So, Jacinta, we want to talk about those. I don't have a lot of information on it, but we definitely want to talk about it a little bit toward the end. But I want to start with some sort of a context setting paper. So, field experimentation enhances translation for behavioral neuroscience. So you say here the brain did not evolve in a lab. It evolved in forests, deserts, and oceans amid predators, rivals, mates, and uncertainty. Yet behavioral neuroscience continues to extract behavioral information from captive animals in socially and ecologically impoverished conditions. Laboratory studies have begun to incorporate more ethological elements, social housing, testing both sexes, naturalistic paradigms, even rewilding animals into semi-natural enclosures. Yet progress in field studies has not kept pace. I often wondered about this, Shelley. I did some work in pharmaceuticals, and we have animal models, as you know, and they are in the lab. Um, Mice, not that many dogs anymore, but other things. And the toxicology studies that we might conduct, um, it's not quite natural in many ways. So, I don't know. I mean, their hormones could be quite different in captivity as opposed to a wild animal. And so, in some sense, we are missing a trick, isn't it? I mean, so so what are you arguing here in this paper?
Yeah, so I guess I can start from a behavioral neuroscience perspective. I mean, I think you're exactly right in what you said, Gil. We've known for a long time that we have difficulty modeling human conditions in the lab, right? And so, one of the classic examples is the forced swim test for depression. Depending on how a rodent behaves when you put them into a tank of water, we can say whether or not they're depressed and whether or not antidepressants will work. And we all know now that while antidepressants work in changing their behavior, it's not a perfect model. It may be capturing some aspects of depression, but it's definitely not a model of depression. And I think now more than ever, it's really important to recognize that, given the tools and technology we have now to probe complex brain behavior relationships and to really look at what's going on into the brain on a cellular and molecular level and relate that to circuits and to behavior is really remarkable right now, but it's not going to advance us until we really know what we're testing in terms of behavior and what we're actually modeling. And so it's a really critical time to pay attention to the foundational theories that kind of
Hmm.
set the stage for behavioral neuroscience in the first place, which we seem to be losing sight of with today's technology. And I'll let Jacinta add her perspective as well.
So, Jacinta, you're looking at it from the field,
Mhm.
um, and there you can actually see how they're living, how they're um, even food. I would imagine is significantly different, right? So, um, and so how do you combine? I mean, you're getting sort of two different types of data on the same models, so to speak, right? So how do you combine these two different data sets?
Yeah, so from the field perspective, we do these long-term studies, these long longitudinal studies of the same animal, and we watch them be born, we watch them grow up, we know their mothers, we watch them migrate, get killed, all the things that go on in an animal's life. And so we're always thinking about not so much what is the current mechanism by which this animal makes this decision, but we're actually interested in what is the life history, the rich life history of this animal that has
Mhm.
produced the brain that makes these decisions. And while, so what really brought Shelley and I together is that I was reading a paper of hers about, I think, what we're going to get into, which is this incentive salience and this reward valuation. And I said, well, what makes an animal go one direction versus another? What makes an animal choose this versus choose that? And Shelly said, we actually don't know. We don't know because of the history going into these animals are often impoverished and they live in, sometimes social cages, but for the most part, sometimes
Hmm.
not. And so really understanding the history behind what produced the brain is kind of what brought us together. So then
Hmm.
we discovered that we could actually do a lot of her testing that she's developed in the laboratory in the wild. And we have this really nice population of monkeys. They've already been
Yeah.
trained to come to platforms to interact with apparati, and we can
Hmm.
now put out the same apparatus that Shelly uses for her rats, we can use for our monkeys. And we're hoping to get an answer to that question. What life history produces the brain that chooses one versus chooses the other? And I expect we'll get into talking about the reward valuation shortly.
Yeah. So I haven't quite thought about this. So there's a longitudinal aspect to it, as you say, Jacinta, that yeah, the history of the animal matters. History of humans. I mean, if some sort of extraterrestrial came here and put us in cages and tried to study our behavior, I think they will get a lot of noise in the
Right.
data, I would imagine. And so same thing is happening to animals. And
And
yeah, go
oh,
ahead.
so
Sorry. Yeah.
we know that an animal that experiences adversity might make very different decisions than an animal that experiences plenty. What's the opposite of adversity?
Advantage.
Advantage. Sorry.
Yeah.
All right, let's cut that one.
No.
An animal that experiences adversity is going to have a very different decision-making process than an animal that experiences advantage.
Hmm.
And that has been shown in humans, it's been shown in all animals where people have looked at it. But to get at this really important decision making, we now want to look at animals who might be low ranking compared to high ranking. Maybe they
Hmm.
lost their mother when they were babies. Maybe they were raised by a really good mother. Maybe they're really well socially integrated versus an animal that's on the periphery. There's all of these different historical things that build brains, make connections in ways that might alter the adaptive decision making. So in both cases, we're going to argue that we think that this decision making is adaptive. It's just that when you've experienced a life of adversity, what's adaptive might be different than if you've experienced a silver spoon type existence.
Hmm. So Shelley, so if you bring, let's say, mice into the lab, they're experiencing, I would imagine, I'm speculating here, some sort of discontinuity in their social structure, a shock, a significant shock. And that should change them too, right? So by the time we start experimenting with them in the lab, they've gone through some massive shock. And it potentially a different animal by the time we get to them.
Yeah, I mean, there's no question that the animals that we're studying in the lab are different than wild mice and wild rats. There's absolutely no question. I mean, in some cases, they're bred and born in our lab in the same environment, in the same cage, experience nothing other than that. In other cases, they're purchased from a vendor. We bring them in, then we do our experiments on them in the lab. Where they come from matters. We've done studies, we've shown that what vendor they come from matters, how they were bred, how they were raised there, what their
Hmm.
conditions are once they get to our lab, whether they're singly housed or housed in pairs or housed in an enriched environment, all of that matters, whether they experience early life adversity. We know that that matters. That matters in terms of behavior and it matters in terms of brain function. But what's different, what's also different from the conditions in the wild is that, especially in mice, they are inbred populations, right? So the genetic variability is squashed in a lot of our populations. And even in
Hmm.
outbred rats, they're not as genetically diverse as we would like them to be. And so that's a whole nother component where you're wanting to look at sort of gene environment interactions. And we're really limited in what we can do in that regard in the lab. And this data set and this opportunity in the wild is just very exciting in that regard.
Yeah, so from I mean, both of your educators, so from an academic perspective, how do you put this together? I mean, is this sort of perhaps a redesign the way that we think about teaching kids? So what do you think about that?
Yeah, I mean, so yeah, I'll go ahead and start, and then Jacinta, you can chime in. I do. I think it's as we kind of talked about in that paper, it's a new way of thinking about our fields and about merging our fields and how important it is to do that right now. And so, yeah, to our knowledge, very few, if any, studies are using wild populations, especially of non-human primates, putting the same apparatus that we use in the lab, that we're kind of considering this not just a behavioral apparatus, but it's a way to peer into the brains of these animals in a non-invasive manner. And so when Jacinta asked me what's going on, what makes an animal, what makes a rat do one thing versus the other, I could tell her in terms of the brain. I could tell her, this is what I think is going on in terms of the brain function and what's driving this behavior versus that. But I couldn't tell her much more than that. And so we know what's going on in the brain in these animals. Like I said, we're lucky to have this technology where we can probe those complex brain behavior relationships. But what we don't know is what's driving that in the first place. So then taking this apparatus as a way to look into the brain of these monkeys in a non-invasive manner, and then use this really rich data set and their really rich environment, and their genetics and their hormones and everything else, and determine what's driving these differences is what's really exciting about it.
Jacinta, what do you think about that as well?
Yeah, so the idea might be to maybe oversimplify. Some brains are very top down, and some brains might be very bottom up. And again, that's an oversimplification, but for the most part, then I care about, well, what makes a top down brain and what makes a bottom up brain? Is it, adversity that makes a top up or bottom up brain, and is it advantage that makes a top down brain? And that might be like the starting point where we frame hypotheses around that kind of basic prediction, and we build from there. And we hope to get at some really nuanced relationships. But for the most part, this assay test that Shelly uses, which is called goal tracking and sign tracking, is really valuable because more so than just, does an animal have behavioral flexibility? Does an animal have a certain level of cognition? This sign tracking and goal tracking test really is a window into the brain in a way that I think a lot of other cognitive tests aren't. And we can't trap our monkeys. We can't look inside their brain. But if we can look at how they make these decisions, we sort of might be able to say that we do know a little bit about their brain based on how they decide to value rewards. And that's really the value. We're bringing a window into the brain, into the wild. And we have full histories on these monkeys. So that part is already in existence. All we need to know is now, how do they make this decision that we know so much about in terms of what brains make which decisions?
Yeah, so there's a sort of a hardware effect. Neuroscientists hate me when I try to make analogies to the computer. There's a hardware effect, and then there's a software effect. One could argue, the difference between natural environment and the lab environment, sort of a software effect. But the hardware, as you say, Jacinta, could have some, they might have IBM chip or Apple chip or whatever, top down or bottom up. So let's go into another paper. I mean, I think it put this in the right context. So you say here leveraging individual differences in cue reward learning to investigate the psychological and neural basis of shared psychiatric symptomatology, the sign tracker, goal tracker model. Yeah, so Shelly, as you know, many of your colleagues have been on before, and we talked about this, but I'm still in the dark, actually. I'm just trying to learn this. So you say here in our modern environment, we are bombarded with stimuli or cues that exert significant influence over our actions. The extent to which such cues attain control over or disrupt goal-directed behavior is dependent on several factors, including one's inherent tendencies. Using a rodent model, we have shown that individuals vary in the value they place on stimuli associated with reward. Some individuals, termed goal trackers, primarily attribute predictive value to reward cues, whereas others, termed sign trackers, attribute predictive and incentive value. So yeah, so let's put this in the sort of a definitional context. So goal trackers, if I don't know anything about this, Shelly, as you know. So goal trackers are basically saying, hey, I'm going to go here and get some food, and I'm not waiting for a bell to ring or a light to flash or anything. But sign trackers have something else that when the bell rings or the light flashes, they sort of know the food is coming. Is that the way to think about it? I mean, I'm making it very crude.
No. Yeah. Let me add a few more layers to it just to
Yeah.
make sure that we're all on the same page. So, the way we classify sign and goal trackers is like you said, we have sort of the classical Pavlovian conditioning, that bell. You can think in this case it's a lever cue, and then the reward, which is a food. And the goal trackers, actually, the bell means something and the lever means something to the goal trackers as well, right? So for goal trackers, it's predictive. It's predictive. They see the lever, they see the cue, and they immediately go to the food cup and they wait for it to be delivered, even though it's not going to be delivered until, in this case, the lever retracts. That lever is a signal for impending delivery. They know that. They assign predictive value to it, and they go to the food cup and they wait for the food to be delivered, which happens right when the lever is retracted. The sign trackers, for them, the cue is also a predictor. They also know that it signifies food reward, but they attribute what we call this extra incentive motivational value to the cue too. So for them, it's not just predictive, but it becomes, in Kent Berridge's terms, this motivational magnet. So for them, the cue itself becomes attractive and irresistible to the extent that they'll work for presentation of the cue, even in the absence of reward. But what's really important about this paradigm is, one, just to reiterate, this is classical Pavlovian conditioning. So the food will always be delivered. The animals don't have to do anything. Food always follows cue presentation. Two, they're both learning something. They're both attributing value to it, and they're doing so at the same rate. We know that both in rodents as well as in humans, that they're able to learn at the same rate. It's the value they're attributing to it that's different. And we know that in rats because of what their response looks like to that cue. So the sign trackers are called sign trackers because they go after that sign, that signal that is signifying reward delivery. And the goal trackers are called goal trackers because they go to the location of the reward, the goal, essentially to consume the reward. And so hopefully that's a little bit more clear in terms of what the topography looks like and what the definition of actual sign and goal trackers are.
Mhm. Yeah, I didn't appreciate this. So the would you say the goal trackers are more planning in the sense that they have to put a couple of things together, right? So there's some sort of lever or light or bell, but then they're not going to go to the sign. They're going where they're going to get the food. So they basically have understood
Yep.
that the sign is different from the food,
Mhm.
or how the food is delivered. But sign trackers are basically just going after the sign, and they also ultimately get the food, but
Yep.
it's really driven by the sign per se.
Yeah. So that is exactly how we think of it. So we think of goal trackers as more sort of cognitively driven. They're thinking
Yeah.
about it. They're thinking about their surrounding environment. They're attributing this predictive value, which we think makes sense. And then they're going and they're waiting for the food to be delivered. And we think of sign trackers as being more kind of emotionally and motivationally driven, without putting much forethought into it, without really engaging that cortex and allowing that inhibitory control. And that's exactly how we think of them, like I said, and we have data to support that that's actually what's going on in terms of the brain mechanisms. And in terms of computational language, we think of sign trackers as relying on what we call model-free learning, and goal trackers relying on model-based learning.
Model-based learning. So Jacinta, when you look at animals in the wild, um, do you have some sort of statistical measure of how many are sort of sign trackers and goal trackers?
Well, we've just started, so we actually don't have, took us several years just to get the apparatus to fit the monkeys and for the monkeys to have a reward that is not going to interfere with their foraging patterns. It's very important that we don't interfere with the monkeys in their natural lives, just from a conservation perspective, as well as an ethical one. But this is the exact question that I came up. This is why I met Shelly, essentially, as I saw a talk on sign tracking and goal tracking, as everyone does at Michigan. And I said, I get goal tracking. No one in their right mind would not understand goal tracking. I don't understand sign tracking. Why? Why would an animal sign track? I don't question that it's adaptive. I'm sure it is. The brain is amazingly plastic, and a lifetime of, or eons and eons of evolution have produced the brains that do these decision making. So I get that it's probably adaptive, but why is it adaptive? And so that was sort of the starting point to our grant and our study together and our paper together, and hopefully many more, is to basically try and figure this out. Is what exactly is it, the hardware or the software? Back to your analogy, is and is it an interaction between the two? So, for example, to use your hardware and software metaphor again, the hardware is the brain, but the life of this animal changes the connections in that brain. And so there can be those kinds of changes are kind of in between software and hardware, I think.
Yes, I'm going to get some hate mail after this. But we have human sign trackers. They're typically found in the financial markets. If you watch the stock markets, for instance, many of the sort of the day traders are sign trackers, actually. And it's very, very similar to mice in many ways. And so, yeah, I mean, we have seen this in a lot of different biological systems that sign tracking is. I'm just trying to get to Shelly. In the lab, do you have a statistical measure? What percentage are sign trackers and what percentage are goal trackers?
Yeah, so we primarily study rats, and so not mice, but focus on rats. And in really large populations, we've generated data sets up to 6,000, 7,000 rats. And when we look at large populations like that, we see that it breaks down into about 30% of the population are sign trackers, 30% are goal trackers, and 30% fall in the middle in what we call intermediate responders. And these are animals that will vacillate between sign and goal tracking, may on one trial go to the lever and on another trial go to the food cup, or do both in a given trial. And what I realized over the years, especially as we started to do pharmacological studies and neurobiological studies, is that I think now that most of us are walking around as intermediate responders. Most of us are able to flexibly adapt depending on the situation and decide, is it better to be a sign tracker right now, or is it better to be a goal tracker? And interestingly, in the lab, when we do these neurobiological manipulations, it's the intermediate responders that we can then push to the extremes. Whereas it's sometimes difficult to break the sign trackers or the goal trackers, but especially the sign trackers. And so, yeah, like I said, in large populations, it seems to, in the lab, it seems to be around a 30% split. But we also know that this varies, as I alluded to before, depending on vendor, where you get the animals from, whether or not they were raised in the lab, whether or not they have a certain transgenic background. So yes, there's a lot of factors coming into play here.
So approximately one third, one third, one third. The one third in the middle. When you say these are the ones you can push to the extremes, what do you mean?
So, we talked about how we think that goal trackers are more kind of cognitively driven and they rely on their cortical control mechanisms, whereas sign trackers rely on more subcortical, motivational, emotional systems in the brain. That's how we think about it. And so when we do certain neurobiological manipulations, for example, now we can turn on that top-down cortical control and see what happens, or turn it off and see what happens. And so we've actually shown that in sign trackers, you can turn on that top-down cortical control mechanism, and now all of a sudden they're not attributing as much incentive motivational value to the cue, and they're behaving more like goal trackers. And in contrast, in goal trackers, you can turn off that inhibitory control, and now all of a sudden they start to behave like sign trackers. And while I say that they start to behave like that is very true, but if we do these manipulations in intermediates, all of a sudden now in intermediates, if we turn on that top-down cortical control, they're definitely a goal tracker. Or if we turn it off, they're definitely a sign tracker. So we're able to push them to extremes with these manipulations, whether neurobiological or pharmacological, much more so than we are if the animals start in the extremes.
Yeah, so they have, I don't think it's the right term, some sort of adaptability or flexibility in the sense that given the environmental condition, they can go one way or the other, whereas original sign trackers or goal trackers may have some rigidity in their brain
Exactly.
that they cannot be really adapted
Yep.
to
Exactly.
the new situation. Um, so, um, so I want to talk about humans a little bit. So I understand that. So you say here sign trackers, the reward cues transformed into an incentive stimulus as capable of eliciting maladaptive behaviors. And obviously this is happening. So let me ask you this. So there are human sign trackers and human goal trackers as well?
Yeah, so there's been a number of studies done where people have looked at this using different approaches. And I'll start with the study that I was involved with, which involved kids. And so we looked at kids that were either 5 to 7 years old or 9 to 13 years old. And what we did is use a very similar apparatus to what we're using in the rats and what we're using in the monkeys. And put it in front of the kids. And what we found was that in that whole age range from 5 to 13, almost all of the kids were skewed towards sign trackers. So we had very few that looked like goal trackers, if any, in each population. And in retrospect, we sat back and we said, okay, so we either design this task completely wrong, and this isn't the right task to capture these individual differences in humans, which may still be true, or it makes perfect sense that we wouldn't be able to see goal tracking in kids of this age because their cortex isn't fully developed, and we know that they're not relying on these executive function mechanisms. And so what was interesting in the study with the 9 to 13-year-olds is that there were a number of things that mapped on really nicely to what we found in the rodents. And so one is we divided them into what we called sign trackers and non-sign trackers because we didn't have goal trackers, but we had a lot of variability that we could play with. And so when we look at sign trackers and non-sign trackers, those that were sign trackers, when we looked at their brains, seemed to rely on these subcortical mechanisms, in this case, the amygdala, to make these cue-elicited decisions. Whereas those that were non-sign trackers relied on the cortex, just like we saw in the rodents for the goal trackers to make these cue-elicited decisions, reward decisions. And interestingly, what I really liked about this study, even though a lot of these assays were from parent report, but what we found was that these kids had tendencies, the kids that had a tendency to sign track also had tendencies for behavioral issues, oppositional defiant behavior, lack of inhibitory control, obsessive compulsive disorder tendencies, and the list goes up, exaggerated fear behaviors, and the list goes on and maps on almost identically to what we've shown in the rodent models. And so we've seen in rats that those with the tendency to sign track also have, enhanced impulsive behavior, enhanced compulsive behavior, more likely to relapse to drug-seeking behavior, exaggerated fear, and similar to post-traumatic stress disorder. Like I said, the list goes on. And so that match between those tendencies in the kids and what we saw in the rodents was really interesting and exciting and really validated what we've been doing in the rodents thus far. And in adults, like I said, there's been a number of other studies, some using tasks similar to what we use in the kids, some using computational tasks. And what's really consistent across all studies is that this tendency to show impulsive behavior in sign trackers has been shown in almost every single study that's looked at sign tracking behavior in humans, and also these sort of reliant on these subcortical mechanisms, brain mechanisms as well.
So this could be potentially an early diagnostic measure. So you say here neurobehavioral profile of sign trackers is relevant to several psychiatric disorders, including substance use disorder, impulse control disorders, obsessive compulsive disorder, attention deficit hyperactivity disorder, and post-traumatic stress disorder. I know in the larger population, these disorders are sort of appears to be increasing. I don't have any data on it, I mean, at least anecdotally. And so, but let me ask you, so loaded question. So 9 to 13-year-olds, yes, so you say, well, their brain is not fully formed. They're still, sort of learning. And as you say, Jacinta, it's sort of a bottom-up brain, so to speak. And they get to this sign tracking thing. But if you look at longitudinally, do they sort of switch over at some point? At what age do they switch
Yeah.
over?
Yeah, so that's hope. That's our hope, and our goal is to do longitudinal studies in these kids, and we hope to actually follow up the ones that we've already looked at in 9 to 13-year-olds are part of a larger study, as well as their siblings. But we haven't done that yet. But that's exactly right. Is hopefully someday this would allow us to be able to predict who may be more or less susceptible to develop these psychiatric conditions and help, kind of, get in there ahead of time to prevent these from becoming disorders in these individuals.
Mm. Yeah, I mean, it's almost like the diagnostic parameter could be the slope, assuming that,
Mm-hmm.
Things are going to change over at some
Right.
point. And if they're not changing over fast enough, it's sort of a, what do we call it? Like early learning. Uh, if the kid is not talking,
Mm-hmm.
when
Like
they, when,
interventions and yeah.
yeah. So that's sort of a similar concept here, right? Isn't it?
Yep.
I mean, yeah.
Yeah. So we don't know yet what that sort of trajectory should look like, and then what the predictive trajectory looks like. But that's exactly what our goal is, to be able to identify those.
Yeah, it's
And just to
fascinating.
go
Yeah.
back, sorry,
Yeah. Jacinta, yeah.
sorry to interrupt.
Sorry, yeah.
Going back to what we opened with the rats being in cages in kind of impoverished life, they have a much more impoverished life, so it's much more difficult to kind of assess what produced this brain. In humans, we
Yeah.
have the opposite problem. We see that they're sign trackers or goal trackers or non-sign trackers, but their lives are so complex, it's really hard to simplify it down to something. So the hope with the monkeys in the wild is really to merge those two. So we have a simpler life. These monkeys that really do just they all move around together. They all kind of eat the same food. They have rank, and they have, their mom is alive or their mom is not alive. They have a social network or a poor social network. And so we can kind of measure these variables much more easily and look at then whether they're a sign tracker or a goal tracker or somewhere in between.
Yeah, that's powerful. So let me make an argument. You guys obviously can object to it. Humans are in the lab. I mean, if you want humans in their natural habitat, you have to rewind time 100,000 years. And so we put them in this houses, schools, vehicles. You sit there and drive the thing for 3 hours to go to work. These are all labs. I mean, these are not natural Homo sapien environments. And hence, we don't have an alternative, sort of a natural Homo sapien experiment going on. I don't know. I mean, you do some work. We will get into this, Jacinta. You did some work in Ethiopia, you're doing some things in Costa Rica. Um, if you go to less developed countries, um, are we do we have different type of data? I'm thinking about Homo sapiens right now, but we'll get into your subjects. Do you think we'll get more natural data from less developed countries?
Well, it's important to understand, obviously, all humans have evolved for the same amount of time. So in that sense, it could be that we're bombarded with unnatural stressors that didn't shape our current existence. And people make this argument about the stress response a lot in saying that, currently we have traffic jams and mortgages and a boss that we don't like. And these are real stressors. They really do affect our blood pressure and things like that. And so they affect our health, but they're not affecting our survival, at least not
Yeah.
in the same sense that like a lion chasing us would. So in that sense, you might say that we're mismatched. We're mismatched with the environment. And so I think your question then is asking, well, could we go to, maybe a natural birth population or a population that has less access to modern inventions? Are we getting to something more equivalent to maybe what early Homo sapiens evolved under? And the answer is probably. Certainly, if we're trying to understand evolutionary pressures, we do want to look at more natural birth populations to look at what affects reproduction and survival. And if we are looking at, sorry, I lost my thought.
Yeah, I mean,
You can
that's
edit.
very.
It's okay, right? It's easy to edit.
No worries. I mean, that's a thought, right? So this may not be a good example, but I grew up in South India. When I was growing up, in the 80s. It was not well developed. Um, it was organized very differently. Now, my parents still live there, so when I go back now, these are big cities. I mean, these are very different way of living anymore, and this all happened in like 40 years. So it's as a very much like getting a mouse from the from the wild and putting that mouse in a cage. We took Homo sapiens from the wild and put them in a cage last 30, 40, 50 years. And that is sort of a natural experiment, isn't it? I mean, there should be some data there that could be interesting.
We're sort of looking at our decision making, even though we're looking at how we make decisions now, our brains evolved, hundreds of thousands of years ago, and there really isn't a whole lot of brain size evolution in the last 200,000 years, so if you're trying to
it's actually brain size is declining after ChatGPT
yeah
showed up.
I'm sure in teenagers. So the brain itself is, we're looking at how we made decisions at a time when we were threatened, our survival was threatened, and so it's kind of almost in some sense a window into our past decision making, or at least what would have been adaptive in the, with an ancestor that was probably much more similar to a chimpanzee.
Yeah, so I want to go into your paper also, Jacinta. So information ecology, you say here, an integrative framework for studying animal behavior. You say here information is simultaneously a valuable resource for animals and a tractable variable for researchers. We propose the name information ecology to describe research focused on how individual animals use information to enhance fitness. So you're doing some very, very interesting experiments in the wild. Um, and the Simien Mountains Gelada research project in Ethiopia, you said um, it's sort of in dormant condition now. But the white-faced Capuchins at Taboga Project in Costa Rica, that's something that you're actively involved in right now?
Yes, that's right. We have several projects going on, and one of them is the project with Shelly Flagel on the sign tracking and goal tracking.
So you are doing sign tracking, goal tracking type experiments in wild Capuchin monkeys?
That's right. So we have a box. It's very similar to the one that Shelley uses on rats, and a lever. The monkeys approach the box, and as I said, they've been trained to come to these platforms. Not trained. We just have habituated them to it. They voluntarily approach the platform, and if they basically break the motion sensor, the lever comes out, and when the lever goes back in, they get a reward. And then they basically can continue. We have several cameras on them and try and score how much they interact with the sign versus how much they interact with the goal.
And what do you find so far?
Well, so far we're just getting the data up and running. So if you have us on in maybe a year, we'll have a little bit more information to tell you. We do see evidence of both sign and goal trackers. Like we've had monkeys go to the sign and we've had monkeys go to the goal. They haven't been completely trained yet, in the sense that I don't know where they have to learn the task. They basically learn that the sign means the goal, and having the same monkey come over and over again, sometimes that takes 25, 40 trials before we call them kind of, okay, now they've experienced both sides enough. Now let's start our data collection. So that's at the point where we start to measure them as goal trackers or sign trackers. And we haven't gotten to that point yet with a monkey, as we get up and running with not just getting them to come to the boxes, but also getting AI to recognize their faces at the boxes so that it can be an automated system. We have these smart systems that will run even when we're not there.
So question for both of you. So is there some sort of genetic component here, Shelly? I'm thinking neuroscience from a hardware perspective, but also culturally on the software side. So if my grandmother were a sign tracker, what's the probability that I would turn out to be a sign tracker as well? I mean, there's a bit of training involved here, right? I mean, it is coaching information passed on to the next generation in some way. So do you see any evidence for that?
Yeah, so it's definitely heritable. So we know that. We know that from studies where we've looked at these traits in rats that were selectively bred for a different trait. And we found that we happen to co-select for the sign and goal tracking tendencies. And so we could predict with 99.9% accuracy
Hmm.
based on how they responded on this other trait. In this case, it was locomotor response and novelty, whether or not an animal was going to be a sign or goal tracker. We also know that I've mentioned a few times that certain vendors and certain barrier rooms from certain vendors will give us all sign trackers or all goal trackers that we assume is due to the breeding as well. And we know in my lab that certain transgenic lines, even when we pay attention to who's a sign or goal tracker, it's really hard to kind of break that, and we're producing more and more sign trackers and more goal trackers. And so we know there's a heritable component. In terms of genetic markers, we have done studies to look at that in the rats, and there haven't been many that have come out. But then, again, in retrospect, why would we expect, one genetic marker to, signify this complex trait? So
Hmm.
it is heritable. There's no question. In rodents, we've seen that. But we've yet to sort of identify the genetic markers that it travels on.
Yeah, I remember you mentioning in the paper, I didn't read it fully, but so I think Jacinta mentioned this too. There is sort of a bottom up and top down sort of thought process.
Mm-hmm.
And it appears that the subcortical structures are more active in the sign tracking, and cortical structures are more active in goal tracking. So that sort of implies sort of brain structure determines who you're going to be. So that could be so the inheritance part of it seems makes sense. But I also wondered there has to be some cultural aspect to it too, right? I mean, there is I mean, if my mother taught me and my brain is top down and her brother is bottom up, her brain is bottom up, let's say, but she taught me to sign track, I will sign track, I would imagine, right? So can we tease this out, this hardware versus software effect in some way? And it's both in the lab and in the wild, Jacinta. So it'll be an interesting thing, yeah.
Yeah, I mean, I think, in the lab, we purposely expose them to relatively few trials to identify them as sign or goal trackers. And so, yes, there's learning involved, but we're not overtraining. We're not creating sign and goal trackers. We think we're really capturing that sort of inherent tendency.
Yeah.
I mean, it's a really interesting question, and it's something in the monkeys that we'll be able to answer is if another monkey sees the animal interact with the lever, does that mean they're more likely to interact with the lever? And so that's something that's going to be really interesting. And something that we've seen so far is, when one monkey comes and interacts, there tends to be a few monkeys around and some monkeys looking from the tops of the trees. And so we're really
Mm.
curious about answering that question, and I think we'll be able to in the wild. And I think when you think of humans and you think of sort of diagnostic ability and things like that, that's exactly what we want to do is if you think of this in terms of top down and bottom up, hopefully we can teach people. And there's this cognitive behavioral therapy that's going on now, like
Mm.
engage your cortex. And I tell that to my teenage son all the time before he goes out. Engage your cortex, be sure you think things through, make good decisions. And so hopefully we'll be able to train people, if we know that they have that bottom up tendency, that they need to
Mm.
start relying more on their cortical control.
So Jacinta, so the Capuchin monkeys, um, as you say, they're very intelligent animals. But are they social? Are they, do they have sort of community and they learn from each other and all of that?
Yeah, they're notorious social learners, and they make really good subjects for this kind of study. So, for example, you can have asocial learning, even if you live in a social group. One animal is playing with something and you approach and start playing with it as well. You learn how it works just because you're attracted to that individual, but you learn individually. But if I'm watching that individual and I learn how to play with it the same way that they're playing with it because I'm watching them, that's social learning. So there is a whole social learning component in this as well. And you might want to think of it as there's 3 different ways an animal could be a sign tracker or goal tracker. One is that they learn, they watch another animal and do that. Another way is that there is a nurture component of the environment. So something in their early life history, maybe they just experienced a really bad day, the current day, that changed how their current hormones and neurophysiology is working, and they're just more a little bit less cortical and more subcortical. It could be something that happened in the last week. So there could be this lifetime change, whether it's early life, the last week, or the last hour, and that can alter how they change, whether they are sign tracker or goal tracker, or it's genetic in the sense that they're hardwired and they're going to be a sign tracker no matter what we do. We know that's not true because Shelly has been able to flip them from sign tracking to goal tracking. So that is, even though there is probably genetic components, it's not hardwired in this instinct kind of way. And so now we can get at that second question, which is how much of it is their lifetime, construction, the hardware basically that's been built in their brains, or how much of it is just cultural. They're learning from their neighbor.
Yeah. So the interesting question for me is, Shelly, so can you really switch a sign tracker to goal tracker? What I'm thinking is that you say 1/3 of them are in the middle. They have adaptable, flexible brains. You can clearly push them to the left or the right. I may put it that way. But can you really take somebody on the left and make that person a right person?
Yeah, so in rats, we can't. We can take them from the left and we can push them to sort of the right of the middle. And we can take somebody from the right and push them to the left of the middle, but we've not yet been able to push them from one extreme to the other extreme. And I think that's actually a function of our manipulations. And so I have this kind of dream experiment that I want to do where I call it a push-pull experiment, where I'm both, simultaneously turning on that top-down control and turning off that subcortical drive. And then I could probably make a sign tracker or a goal tracker. Or in contrast, I can turn off that cortical control and turn on that subcortical hyperactivity and then make a goal tracker, a sign tracker. But I think just, the way we've been doing it, where we're administering a drug systemically or turning off one pathway or turning on another pathway, that's not going to do it. We need to do more than that. And that's, the brain is complex, and we should have realized that in the first place. But it is difficult to make from one extreme to the next.
Yeah, so sort of an extreme view. So if you say psychiatric disorders that we talked about have a higher correlation with sign tracking behavior, let's say, then the therapeutic interventions in the middle will have higher value. Therapeutic interventions in the extreme probably have less value in some way. Just making a statement. I haven't really thought about it.
Yeah, I mean, it's possible, but it's also, goes back to this early intervention idea. So if we can catch them before they're stuck in the extremes and then do the interventions at that time, maybe we'd have better luck. But as you know, in terms of psychiatric research and neuropsychopharmacology, it's not been a hugely successful field for various reasons. And I think one of those is actually this notion of individual differences, and that we haven't paid attention to it much. So I think using the sign and goal tracker model in that regard, just to identify who may be more or less prone for different reasons, is really a step in the right direction.
And the dominant position to be, sounds to me, is in the middle. Because you have more flexibility, more adaptability, both from a disease perspective as well as from a sort of objective function perspective. So then that goes into, let me ask you this, I'm stretching this a little bit, into education. So we take kids early, let's say, can we design education systems in such a way that they are sort of in the middle, gives them more flexibility, more adaptability?
Yeah, I mean, so one of my fears, and no real evidence for this, but is that, we are with kids and with phones and computers and technology in their face in the way that they're growing up. So you talked about, you growing up in us growing up in the 80s and how that's changed, but now look at the kids today and what they're actually exposed to, and a lab environment that they're stuck in their phones and their devices. And that could be making them, they have a really hard time, and I see this like engaging their cortex, right? And so they're just
Mm,
constantly scrolling, not paying much attention, not really even registering what they're looking at, and just having these actions just sort of consistently without much forethought. And so
mm,
I think that yes, I think we do have to teach them to engage their cortex and take different approaches to things and rely on different parts of their brain, and kind of what's going to become the default. And I think it's going to, kids' brains now probably look very different than they were 10, 20, 30 years ago, and it's going to keep
mm.
changing.
And how do you engage the cortex? I mean, even undergraduates coming to Ann Arbor and you design a course, what are the techniques you can use to engage their cortex rather than subcortex?
Yeah, that's a great question. I'll let Jacinta chime in terms of the educational component. I'll say from the psychiatric angle of things, like I said, there's cognitive behavioral therapy going on now, where you can teach people to move an object on a screen by engaging their cortex. And so that's been
Mm,
going on for a number of years now. And so that's a
yeah.
possibility. In terms of education, I'll throw it over to Jacinta and see if she has ideas.
Well, just making them think about things, taking a something that they take as a given and actually having it challenged. And then they have to understand, well, why do I feel this way? And we
Mm,
get that from group learning, small groups, diversity, having a diverse population kind of can help you put yourself in their shoes and wonder why are they thinking that way? They must have gone through a very different experience than I have. And
mm,
you have to kind of think about the decision that you have already made rather than just accepting it that, of course, this is right. Of course, I'm going
mm.
to, do this one thing. And there's lots of pedagogical materials out there that help educators tap into those kinds of teaching skills.
Do you see any sort of discipline bias? What I'm thinking is that, so you think about engineering and medicine, for instance, it's fairly prescriptive. And I wondered if there is some sort of correlation how certain professionals think or do things. I mean, one of our greatest strengths have been, I would argue, the liberal arts education that pushes people in all sorts of different directions. When you push people in all sorts of different directions, you have a more flexible brain, and that has actually therapeutic effects. It seems to me, good therapeutic effects. So I wondered if there is a discipline-related correlation. Have you found any?
Yeah, no, we haven't looked at that, and I don't know of anyone that's looked at that. I mean, I'm sure that there have been studies in terms of brain function and even brain structure, and what that might look like in different disciplines. I know, there's the classic study of taxi drivers in London who have really big hippocampus because they have to navigate the streets of London. That's one of the classic studies, and I'm sure there have been
Oh
studies
yeah.
like that done with other disciplines. I'm just not aware of what they are. But yeah, I mean, I would agree with you that that's definitely the case, and that liberal arts education is very beneficial in that regard.
And going back to Costa Rica, Jacinta, so we'll conclude on this. So I know that you just started the sort of looking at the data. And what's the population that you're looking at? What is sort of approximately what's the population of the monkeys that you're looking at?
It's a really small forest fragment, but there are 12 groups living inside this very small fragment. And the reason that there are so many monkeys in this forest is because it is surrounded by sugarcane, it's surrounded by mango plantations, it's surrounded by bananas. There are oil palm fruits where they get this high
Mm,
calorie fat fruit. So these monkeys are really well fed, and there's also water canals that run through the forest in order to
mm,
bring water to all the crops. So because of the human crops on all sides, the density of the monkeys is really, really high. Now, from an ecological perspective, there's all kinds of things you might argue against, like, oh, they're not living naturally. Oh, they're not living natural, they're not eating all these natural foods. But from our perspective, it is a perfect laboratory because they are so dense. They live in these dense populations of monkeys where, I study social behavior. And so if you want to study like social behavior, you want lots of social interactions. And by having them live in this very dense population, we get all kinds of social interactions, all kinds of fights and intergroup encounters, and even predator encounters. We have predators in the forest as well. So it's a really rich environment we've basically turned up the social volume on all of these interactions. And the whole population is probably about 250 monkeys. We don't know.
Oh,
we
wow.
don't.
Yeah, that's a big population. Yeah.
And it's actually, if you think about a laboratory, most laboratories have maybe 30 primates that they can work with, and that's a big population. A lot of people have 2 or 3 or 4 monkeys, and so you can sort of test for monkeys and publish what they have done in the laboratory. But this gives us this natural laboratory where we don't have to feed them, and we don't have to care for them, and we don't have to the thing we worry about a little is the ethics of our interactions with them. We try, obviously we don't interact with them. We put apparatuses up when we're not there, so the monkeys come and it's already there. So we do take pains to make sure that we don't interfere with what the monkeys would do normally. But for the most part, they go about their lives. They live, they die, they reproduce, they survive, and we can then watch how their decisions play into how well they do all these things.
So you're taking videos of them coming to the box?
Yes. In fact, every box has 4 cameras on it. One for the face to get facial recognition, one for above to get, we use AI also to score whether they're sign trackers or goal trackers. We have one from the side in case one of the side, this one doesn't work. And then we have one way back in the back to kind of get at who else is around and who else is watching and things like that.
Excellent. Yeah, this has been great, guys. Thanks so much for coming back, Shelly. And nice talking to Jacinta.
Yeah, thanks so much for having us.
It was a pleasure to be here. Really, thanks.
Bye.
Thanks. Take care.